Okay, maybe it's 11:01. I guess we can get started. So welcome everybody to our PAS plenary session this morning. This session is on how social determinants meet precision medicine. I just want to make a few comments before I turn it over to Dr. Mike O'Shea. Pediatric Research, the official journal for the American Pediatric Society, the European Society for Pediatric Research, and the Society for Pediatric Research is sponsoring this plenary. Just so you know a little bit about pediatric research, we publish translational research from basic to clinical to epidemiology, quality improvement, and health services research. And in addition, we've begun publishing family reflections on the type of research that we publish, commentaries that come from you, our readership on the articles that we publish, and get more of your thoughts on these articles. We also are publishing graphic artist renditions of mechanistic pathways that come from the original research that we publish. And just recently, we have kicked off a new type of article in the tradition of narrative medicine. And given the experiences that pediatricians are having during the COVID-19 pandemic, we wish to collect their narrative medicine writings of physicians' experience dealing with the issues that this pandemic has raised for us. So in the near future, we're going to be putting out a call for submissions for COVID-19 related experiences in narrative medicine. So watch for that. And now I'm happy to introduce Dr. Mike O'Shea, who is going to be our introductory speaker and moderate this session. Mike.
Right. Good morning, everyone, and thank you for being here. It's a great pleasure to introduce this session titled The Social Determinants Meets Precision Medicine, and I want to take the opportunity to thank Cynthia Bearer, the discussant for this symposium, for developing the concept for today's session, prompted by a series of papers in Pediatric Research. And as you just heard, she serves as editor for that journal. The papers she noticed focused either on precision medicine, which primarily relies on individual level risk factors such as genetic susceptibility, or focused on social determinants of health, which primarily concern risk factors distributed across residential regions, socioeconomic groups, race, or ethnicity. And a solution-oriented question, which she posed in the description of this symposium, envisions care that takes into account both social determinants as well as individual level factors. So as you can see from this slide, the PubMed entries for these two areas of investigation have increased very dramatically over the last decade. So this morning we will hear from Xiaobin Wang from the Johns Hopkins School of Medicine, a talk about a potential mechanism whereby social determinants might become embedded in the epigenome and thereby transmitted from generation to generation. We'll hear about a study by Jackie Bangma from the University of North Carolina, investigating early life prenatal antecedents of positive health outcomes in a cohort of children born extremely preterm. We'll hear from Heather Burris from the University of Pennsylvania, a very comprehensive look at how precision medicine might help us address Black-white racial disparities in the preterm birth risk. and then Annie Behrens from the University of California, San Francisco will tell us about a tool that might index psychosocial risk and be thereby useful in research studies. And then in the last 10 minutes, Dr. Behr will moderate a discussion. We'd like for you to submit your questions using the chat function. If you could please make sure you're muted, but please submit your questions at any time via the chat function, and then we'll address that. We have left 5 minutes after each presentation for questions and answers. And without further ado, I'm going to turn it over to Dr. Wang.
Can you hear me?
Yes.
Great. Good morning, everyone. It's my great pleasure to participate in this webinar. My topic today is integrate social determinants of health with multi-omics in prospective birth cohort study. In the next 20 minutes, I'd like to start with the background and significance. Then I'll share with you our findings in the Boston birth cohort. And finally, I'll talk briefly about gaps and our future research opportunities. So let's first start with social determinants. According to CDC, social determinants of health are conditions in the places where people live, learn, work, and play that can affect a wide range of health risks and outcomes. Examples are poverty, food insecurity, and stable housing, and stable neighborhood, and safe neighborhood, and toxic stresses. So as you know, in recent years, the AAP has issued a series of policy statements and guidelines on social determinants of health. So here I'd just like to highlight the AAP policy statements on early childhood adversity, toxic stress, and the role of the pediatrician, translating developmental science into lifelong health. So in this statement, the AAP endorses our integrated eco-bio-developmental framework for understanding the promotion of health and the prevention of disease across the lifespan that builds on advances in neuroscience, molecular biology, genomics, and the social sciences. So I'd like to bring you to another forefront that is relevant to today's talk. This brings us back to 70 years ago. The pictured here are two Nobel Prize winners, Watson and Crick. Together, these revealed that DNA structure is a double helix. Their work and others have established that the human genome is the primary carrier of the genetic blueprint, which determines an individual's potential for growth, development, health, and diseases. So this slide explains what we mean multi-omics. So on the left side is the central dogma of molecular biology that was revealed about 60 years ago. So it reveals the key biological process from DNA, the genetic blueprint, to transcription and the translation process to determine protein amino acid sequence and the 3D structures, and ultimately form the clinical phenotype. So if you look at the right side, that's the multi-omics. They are the tools and the technology we have to study corresponding layers of biology, correspondingly, genomics, epigenomics, proteomics, and metabolomics. So what are the potential values to integrate social determinants with multi-omics in prospective birth cohort study? First of all, from a scientific discovery and innovation perspective, it will ensure a strong foundation of basic science and methodology, and it will enhance the scientific rigor and reproducibility. From translational perspectives, it will provide critical needed evidence to inform public policies, social reform, health services, and the clinical and the public health programs. So in the following, so I'll share some of the findings and some of the works we have conducted in the Boston birth cohort. The Boston birth cohort is one of the largest and the longest running urban low-income minority birth cohort in the US, comprising of over currently 8,600 mother-child pairs. Our ongoing investigation focuses on early life social, environmental, nutritional, genetic, epigenetic, and metabolomic factors on a range of pediatric and adult diseases. So I would like to start with a classical example that is the interplay of maternal smoking with genome and epigenome. So as you know, about 12% of US mothers smoke during pregnancy, maternal smoking affecting her own health and the health of her baby beginning in utero. The health consequences are numerous. For example, preterm birth, obesity, asthma, neurodevelopmental outcomes. So the questions are, what determines an individual's susceptibility to cigarette smokes? And what is the effect of maternal smoking on fetal DNA methylation? And what factor can counteracting that toxicity? So here's some our early findings. So the left, the panel showing currently is illustrate the metabolic detoxification process for environmental chemical, including cigarette smoke. So so this right panel illustrate the joint association of maternal cigarette smoking and the metabolic detoxification genotypes with preterm birth. So if you look at the right panel, the first red bar is the conventional epidemiological study, result considering genotype of the mother. So the odds ratio, as you expected, it's commonly published in the literature, is about odds ratio of 2. However, when we further considered maternal genotypes, if you see the last bar, that's the subset of the mothers who smoke, also have C1P1E1 and GSTTT gene variants. So this subset of mothers has exceptional high risk of preterm birth associated with smoking. The odds ratio is about 10. So what this study demonstrates is that maternal cigarette smoking, one, can interacting with maternal gene variants to moderate the risk of a preterm birth. Importantly, we found a subset of mother appear to be at a particular high risk of preterm birth because they're gene variants and can interact with the cigarette smoke to increase the risk. So this subset represent a high-risk group and could form the basis for target intervention. So here it's further studies we look at, can maternal smoking affect the fetal DNA methylation? And can adequate maternal folate counteracting the risk? So in this analysis, we included both mothers, African American mothers, and the children from the Boston birth cohort. So I'd like to orientate you a little bit about this slide. The y-axis is the difference in DNA methylation between smoking exposed versus unexposed children. So what we present is the difference. If there's no difference, it should be around 0. And if it's increased, demethylation should go up, and if it's decreased, demethylation should go down. So as you can see for the CPG sites, that's the loci, gene loci we found that has altered demethylation. Smoking in general tended to decrease the amount of DNA methylation. So another thing I'd like to draw your attention is, in addition to related to decreased demethylation, here has a gray bar and the black bar for giving CPG site. So what this means is the gray bar is the children born to mother with low folate, and the dark bar is the children born to the mother with high folate. As you can see, maternal high folate appear to attenuate the effect of smoking on fetal DNA methylation. So here I'd like to show you another interesting findings we revealed. Is if you look at the top one, it's for the overall sample. If we look at the bottom one, that's for the males only. So you can see the pattern is similar, but the effect, the size for males is bigger. So to sum up, what this study demonstrated is that maternal cigarette smoke can affect fetal DNA methylation. That's one. And second, the male fetus appear to be more susceptible to maternal smoking effect on DNA methylation. And lastly, also very important, is this is the first time we demonstrated maternal adequate folate level actually can counteract smoking toxicity. So I'd like to show you another example, which is related to maternal stress and the social support on DNA methylation. So in the Boston birth cohort, overall, we found 14% of mothers reported experiencing high stress during pregnancy, and the rate went up to 24% among mothers who also reported lack of a baby's father support, and up to 38% among mothers who were lack of a family and friend support. Concurrently, the other studies, previous studies, showed that the perceived psychosocial stress can increase the risk of adverse health outcomes, while social support can promote resilience or indirectly buffer stress during pregnancy. So the question we hope to ask is, can psychosocial stress or social support affect maternal DNA methylation? And this is especially important among African Americans who are disproportionately affected by psychosocial stress and the lack of social support. So in this study, we analyzed DNA methylations among 250 African American mothers. So our stress and the social support were based on maternal questionnaire interview at the enrollment. So this is, sorry, this is a little busy, but I'm gonna walk you through. So this is actually the main finding of this study. So as shown in this table, lack of support from baby's father and the lack of support from family or friends were associated with altered maternal DNA methylation at multiple CPG sites. These findings, although still preliminary, if further confirmed, would provide evidence for biological embedding of social support and may serve as evidence for strengthening social support as a strategy to improve maternal health outcomes during pregnancy and perhaps beyond. So here are some perspectives we learned through this process. What we have done so far are quite preliminary and mainly hypothesis generating. And there's many gaps remain in the field. For example, although the importance of social determinants and the genetic susceptibility in health and disease are well recognized, few studies considered both in the same study. The existing study even bring these two together. As example, what we did relatively small in sample size, and a few has been replicated. And also few studies were conducted in longitudinal birth cohort. And also there is a particular lack of studies that were conducted in disadvantaged US minority mothers and children. As we all know, they bear a disproportionately high burden of social adversities and disparate health. So I'd like to end my talk with this slides highlighting our team's aspiration and effort to connect multi-level, multi-dimensional social determinants of health with multi-omics to promote health and prevent disease across the lifespan and the generations. Given the growing recognition on the importance of social determinants, the rapid advancement of biomedical sciences and technology, and the growing number of prospective birth cohort studies, I believe we have unprecedented opportunity to advance this field. So I'd like to take a moment to acknowledge all the contributors to our work. The Boston Birth Cohort has been in operation for more than 20 years. It is indeed impossible for me to list all the people I'm grateful, thankful. So I'm deeply indebted for a number of funding agencies, institutions, and hundreds of people who I have had the privilege to work with and who have contributed to the establishment of the Boston Birth Cohort, as well as numerous scientific discoveries and publications derived from this cohort. Last but not the least, I'd like to thank the organizers of this webinar, Cynthia and Lauren, and thanks Mike for moderating. And thank you all for your time and attention. And I'm happy to answer any question you may have. So I'm gonna stop sharing.
Yeah, thank you so much, Dr. Wang. There are some questions in the chat box that we could take. We've got about 5 minutes.
Sure.
First, does high folate with decreased demethylation mean that just substrate deficiency influences demethylation? And then has anyone looked at high and low choline levels?
And can I just clarify, is this question related to the amount of folate or the type of folate? Let's see, can I look at the chat?
That was my question. So you showed a slide where you had less demethylation when you had high folate. And so I'm wondering if just a deficiency of folate would result in, or higher folate means you have more substrate, more 1-methyl donors. And is that the difference that you're seeing then? It's an unregulated amount of substrate that you can use to methylate your DNA. And then choline is a 1-methyl donor also, and most I guess 40% of the population is choline deficient, and I wondered if you'd looked at choline levels also in your cohort.
This is a great question. As you know, folate is only one member of the one carbon metabolism. There's B12, B6, and a whole bunch of enzymes and the regulate the process, and the homocysteine pathway, DNA methylation pathway, and the synthesis pathway. It's all connected, in homocysteine pathway, all connected in this loop. So what we look at is only the maternal folate level, and this is not only choline may also contribute to the process and also the type of folate. So for example, it's synthesized folate versus a natural folate. So that's why there's so many questions we haven't answered, we haven't studied. So we're hoping, so that's also the excitement keep us going is we found one thing, we open the question for another 100 questions. And this is definitely something we hope either our group or other people can continue to pursue. And the folate, the cholins, beta, for example, all those related to one carbon. And also not a lot of genes involved in the loop. For example, MTHFR and FDR, those genes involved in the loop.
Dr. Wang, another question is, have you evaluated gene-environment interactions such as you found for tobacco smoke related to other exposures such as air pollution, to see if there are certain
That's a great question. And so, no, we haven't analyzed the air pollution and the gene interaction, because I would love someone to do this. I don't know who raised this question. Maybe send me email connecting, maybe we can work together on this. It's just we have over the years, we have been very busy collecting data and has a limited personnel, so we only get too much work done. And certainly, I think it's come to a point, there's a lot of questions remains, and if you're interested, contact me to see whether we can figure out something, work on this question. I think this is a very, very relevant question, important question.
And then another question, your data suggests that paternal support differs from friends and family support. In what way do you feel the support is different and why does it result in different methylation patterns?
That's another great question. I know this is the excitement about science, so we'll never stop. First of all, I would say in the Boston birth cohort, in terms how we measure stress, I'm sure today we have other experts who are true experts in stress measurements. And so we have to say our evaluation of the stress in the Boston birth cohort are relatively crude. And so this is based on maternal self-report. So as you know, anything based on self-report may subject to variations and probably imprecise. And so why some of the sites are related to the paternal stress, and why some of the, paternal support, why some of the sites relate to friends and family. I think that's a great question. So we don't have an answer, but those certainly, I think what we found is very preliminary, hypothesis generating. I was hoping probably we can pursue the following. First of all, we need a larger sample size. I think our sample size is relatively small. It could be due to random variations. So the results may or may not robust. Secondly, our finding hasn't been replicated. So if the finding can be further confirmed and replicated, if we can still see the differences between father's support and the family's support, so that may prompt another level of investigation.
So thank you so much, Dr. Wang. And I was going to ask all of the speakers if you are willing to share your email address. If you could put it in the chat box, then some of the people who are on the call who might want to contact you could do so if you're willing to share your email address. But thank you very much. And we're going to move on to our next talk by Jackie Bangma from the University of North Carolina. Her talk is on the early life antecedents of positive child health among 10-year-old children born extremely preterm. And Jackie, if you'll share your screen, I'll stop talking.
I can do that. Well, thank you, Mike, for the introduction. And thank you, everyone, for joining in today. Today, I am going to be talking about the antecedents associated with positive child health, but I'm also going to be talking broader about the developmental origins of positive child health in children born extremely preterm. So the developmental origins of health and disease, or DOHAD hypothesis, as I will be referring to from here on out, main hypothesis is that exposures during early development are linked to or can lead to adverse health outcomes, either in childhood or later in life. And the exposures in DOHAD could be anywhere from maternal stress to metals in maternal circulation, pesticides, among many other things. Here is just one example of the many types of articles that kind of focus around a DOHAD type investigation with looking at prenatal maternal obesity and how it might be associated with neurodevelopment and executive functioning in children later in life. Now, DOHAD is a relatively established and accepted way of investigating adverse health outcomes, where the adverse outcome is the outcome of interest for a very good reason, in that you can, when understanding what exposes might increase risk or lead to these adverse outcomes, we can change those exposures and reduce that risk. But we can also look at the flip side of the DOHAD hypothesis, where the healthy outcomes are the outcomes of interest. And that is an area that our laboratory has been interested in and starting to focus on. And the field is known as positive child health. And this is a growing field that is supported by the NIH and is a part of the ECHO program. And specifically, our lab focuses on the placenta as a modulator of those exposures and can link to those later in life outcomes, whether adverse or positive. So our lab is currently writing a review and looking at identifying things early in life and risk factors and exposures and how they're linked to both early in life outcomes and later in life outcomes, with a little bit of a focus on positive child health and those later in life health outcomes. So keep an eye out for that. For the studies that I will be talking about today, we are privileged to be able to work with the Extremely Low Gestational Age Newborn Study, or the ELGAN study. And this cohort has a cohort of children who are all born prior to 28 weeks gestation between the years 2002 and 2004. If you can see on our map, our children were recruited in 14 hospitals across 5 states and range between 23 weeks gestation to 27 weeks gestation, as you can see in our pie chart here. In addition, our mothers were interviewed upon an initial enrollment when the children were born, and then were followed up at both 2 and 10 years of age. Currently, we do have a 15-year of age assessment underway, but today I will be focusing on our 10-year assessment, where the children were assessed for a total of 11 adverse health outcomes and also assessed for a quality of life score. In addition, at birth, the placenta was collected for a number of the children, and so that they were able to be analyzed for DNA methylation and mRNA expression. Now, this cohort is an extremely preterm cohort, and so they are at risk for a variety of adverse health outcomes, more so than the general population. So you can see here our Y axis is our percent prevalence in the population, and across the bottom are adverse health outcomes. CP is cerebral palsy, ASD, autism spectrum disorder, and you can see the blue bars represent the LGAN cohort, tend to be higher than our general population in red, with the exception of obesity for those displayed here. And despite the fact that these children are at risk for a lot of adverse health outcomes compared to the general population, they do thrive in many instances as they grow up and in their childhood. And so it is this success and thriving that is the central focus for the positive child health that we are interested in our laboratory, or in our group. So before we get too much further, I'll go ahead and define positive child health, since it is a new and quickly developing field. The positive child health is defined as a state of complete physical, mental, and social well-being as defined by the World Health Organization. It is the extent to which individual children are able to develop and realize their individual potential, satisfy their specific needs, and interact successfully with their environments, both physical, social, and biological. So the children in our cohort who experience these early life adversities, those that have the best outcome, the most positive outcomes, what factors predict these improved outcomes? That's what we're most interested in. And we do have a definition of positive child health and a cohort of children that seem to thrive despite adversity. However, assessing positive child health as an outcome is a relatively new method, and researchers have not quite converged around a definition of operational construct for positive child health. So our goal going forward is to begin to operationalize this assessment and create a tool to understand positive child health. So the aims for our studies that I'll talk about today are to develop a tool using our existing LGAN data, begin to apply that tool and begin to identify pre and perinatal antecedents that are associated with improved health outcomes at 10 years of age, and then also use that tool and dig a little deeper into mechanisms and potentially identify methylation and expression in the placenta at birth that is associated with improved health outcomes at 10 years of age. So let's go ahead and look at the tool. So this study was completed and published in 2018, and we were able to create the Positive Child Health Index, and it is from the 10-year follow-up with 11 adverse health outcomes, as you can see listed here, which our children were assessed for. We wanted the positive child health score, a high score, to represent a good score. So children with no disorders were assigned a PCHI of 100, while any additional diagnosed disorder dropped the PCHI incrementally. And 32% of our cohort actually had a PCHI of 100 at 10 years of age, which is really great. And you can see here, this is our PCHI scores in our cohort by number of children. And so across the bottom on the left, we have our highest PCHI scores, and you go across to the right, the PCHI score drops. And out on the farthest right, we have only one child who was diagnosed with 8 of the 11 disorders. We did see a sex-based difference in this analysis, where our female children tended to have higher PCHI than the male children. Once we had our PCHI, we wanted to ensure that it was capturing a quality of life type information. And so we used the quality of life score known as the PeachQL that has 5 subsections, and altogether those create the PeachQL total. So those created our QRL measure, which we then use a linear regression model to compare to our PCHI. And we did so in several ways. We looked at both binary categorization of our PCHI and a categorical variation of our PCHI. I will only show our p-values for our binary in interest of time today, but you can refer to our paper, or we can chat if you're interested more. The findings between the two models were very similar. And so here you can see our p-values for our binary model, and green represents significant values, And as you can see across the board, our PCHI and quality of life measure were highly similar and seem to capture similar information. So now that we have a tool to begin to assess our positive child health, we want to go ahead and start to identify pre and perinatal antecedents that are associated with improved health outcomes at 10 years of age. So this study was completed and published in 2019, but for this study, we were able to utilize both the structured questionnaire at enrollment and the medical chart to obtain 58 total maternal antecedents. And for this study, linear regression models were again utilized to compare our antecedents and our PCHI. We did both binary and categorical models, which results are combined here. There's more specifics in the publication, but these models were adjusted for covariates and was also stratified by race when we did find a significant interaction value by race. So for antecedents that were associated with our improved PCHI, we saw a variety of health outcomes. We saw that mothers being treated for urine, bladder, or kidney infection, cervical seclause, multiple gestation were associated with improved PCHI. We also saw in our stratified by race analysis, we saw that mothers of Black race with assisted reproduction and proteinuria during pregnancy saw associations with improved PCHI, and then among participants of white race, that receipts of antibiotics during pregnancy were associated with improved PCHI. Now, at first glance, these results don't seem to have a whole lot in common. However, authors feel that this may be a representation or a theme of mothers having high access to high specialized, high quality healthcare. So a mother being able to receive cervical seclage, for example, a mother wouldn't be able to receive that without access to high quality healthcare. Same with assisted reproduction or receiving antibiotics or being treated for urine, bladder, or kidney infection. Now, for mothers with multiple gestation and proteinuria during pregnancy, those pregnancies tend to be of higher risk and therefore are given additional resources and attention during their pregnancy, and that may be a factor involved in those antecedents being associated here with improved PCHI. Now, we also saw that several antecedents were associated with decreased PCHI, excuse me. And where on our and the screen I just showed you, we saw that access to high quality healthcare was associated with increase. We would seem to see that the loss or lack of high quality healthcare seems to be associated with decreased PCHI. And here I'm just showing you a few to begin with to compare to our previous screen. So mothers with switching from private to public insurance between the child's age 2 and 10 was associated with decreased PCHI. And then mothers who had public insurance throughout the study was also associated with decreased PCHI. In addition to that, we also saw that several other antecedents were associated with decreased PCHI, such as mother obesity pre-pregnancy, maternal asthma and requirement of asthma medications, and maternal consumption of aspirin during pregnancy, and proteinuria during pregnancy for participants of white race. So these additional antecedents seem to suggest that maternal health disorders are largely associated with reduced positive child health at 10 years of age, such as obesity and asthma. So now that we've begun to identify these pre and perinatal antecedents that are associated with improved health outcomes at 10 years of age, we wanted to dig a little deeper and start to identify mechanistic links by identifying methylation expression in the placenta at birth. So a quick introduction to the placenta. The placenta is a really important organ in pregnancy and is responsible for nutrient, gas, and waste exchange between mom and baby, and modulates most fetal exposures, including metals, pesticides, cortisol, and maternal medications. The placenta is comprised of numerous cell types, but some important cell types in the placenta known as trophoblast cells are important for migration and invasion into the uterine wall and is able to establish blood flow to the placenta and nutrients to the fetus. So if migration and invasion of the trophoblast is compromised, it is likely that complications develop and increased risk of adverse health outcomes. So for this, we hypothesize that genes involved in trophoblast invasion and, trophoblast invasion and function will be differentially methylated in children with higher PCHI compared to children with lower PCHI. And in addition, we hypothesize that gene expression of differentially methylated genes in the placenta will be altered to match that methylation change. So for this study, we did sex stratification where females and male placentas were investigated separately, since we do see a sex difference between the functioning of the methylation in the placenta. For females, we had a total of 203 placentas that were investigated at 850,000 CpG methylation sites. And our graphic here is known as a Manhattan plot, where we have our chromosomes along the x-axis, and the color variation is just to help visualize the differences between adjacent chromosomes. Our blue and red line here represent our multiple corrections p-value, and so anything below 0.1 we considered significant, and for females we found a total of 9 significant CpG probes within 8 identified genes in the genome. We did the same analysis for male placentas, where we had 223 male placentas, again investigated for 850,000 CpG methylation sites in the placenta. And for males above our FDR correction line of 0.1, we had 31 significant CpG probes that were within 21 identified genes within the genome. And while these DNA methylation results are very preliminary, and we are currently working on this analysis, I wanted to give you a little sneak peek at some of the things that we are finding. So we have taken these genes identified by CpG methylation and looked at gene expression, and I'm just going to show you the male results here. We have, of the 21 identified genes for males, we had several genes, 3 total pop up and 2 that we think are really interesting and wanted to share, that are associated with our positive child health. And that one of them is the ankyrin repeat domain 11 gene. This gene in the literature has been shown to be differentially methylated in the placenta and is associated with neurodevelopment and behavioral outcomes. In addition, our very low density lipoprotein receptor is one of our genes of interest, and it is shown to be a key player in nutrient transport function in the placenta, and the methylation status and expression is also altered in gestational diabetes and preeclamptic placentas. So this seems to suggest that these genes are not only important in neural development and gestational diabetes and the health of the pregnancy, but also long-term health. So now that we have begun to assess positive child health, how do we think that this knowledge can be used to improve the life course of individuals born extremely preterm? So we have begun to identify early life factors associated with resiliency. In addition, we have begun to understand mechanisms that might link early life factors, risk, and resiliency, but we also want to further, in the future, design interventions to modify these factors, and also, of course, rigorously test those interventions. And the reason for this is because the earlier we can intervene at the potential for adverse, or intervene to put a child on the path to more positive child health, the greater chance for a larger change. So you can see we have our life course across the x-axis and our chronic non-communicable disease risk on our y-axis, and the life course is in blue. And the earlier intercession happens, the greater the potential for the drop in that non-communicable disease risk. So the big picture take home from the studies that I've talked about today are that maternal antecedents and early life factors in our cohort are associated with positive child health outcomes, where maternal access to specialized high quality healthcare seems to be a big factor. And both having that healthcare leads to improved PCHI, and losing that healthcare seems to be associated with decreased PCHI, as well as maternal health disorders seem to be associated with decreased PCHI. We're also beginning to see that placental methylation and mRNA expression is associated with improved PCHI at 10 years of age, and that future interventions could help inform us how we might be able to increase the chance of higher positive child health outcomes in future generations. With that, I would like to acknowledge all of our funding sources. It could not be possible without all of our funding sources and our ALGA and PIs as well. There is quite an army of people without who this project would not be possible. And it's not possible to name them all, but with that, I will take any questions.
Thank you so much, Dr. Bangma. That was excellent. And we've got about 3 or 4 minutes for questions in the chat. Someone is asking, did you adjust for socioeconomic status, medical insurance, prenatal alcohol, or drug use?
So it depends on the study. We did, for socioeconomic status, we used maternal education because we had finer, there were more stages in maternal education, and they were highly associated with socioeconomic status of the mother. And so the maternal education was used as our SES status in both the first and second papers that I talked about. As medical insurance, I'm trying to remember. Mike, do you remember? Say that again?
We did.
We did.
Yeah.
So we did control for medical insurance in our analysis. Prenatal alcohol or drug use, I do not believe we controlled for prenatal alcohol or drug use. It was self-report. And so some mothers did say that I can't remember how many mothers say whether or not they drink alcohol. I know that they self-reported smoking, and most mothers either was just secondhand smoke exposure or non-smoking.
Great. And then is it possible that the reason for preterm birth, that is the cause of the preterm delivery, that could affect DNA methylation patterns? And that it could, and also it could affect the outcomes, and thus be potentially I guess a confounding variable.
That is very possible that socioeconomic status is a confounding variable. We did try to control for it in both of the analysis. However, that doesn't mean that we were able to control completely for it. I do believe it probably does have a large impact on the effects and positive child health. So I think it definitely needs to be continued to look into further.
Yeah, and just to clarify, I think the question was whether or not your group is looking into whether the cause of a preterm delivery, for example, preeclampsia or maternal infection, whether the placental DNA methylation varies by cause of preterm delivery. Is your group doing any work to look at that?
Jackie, we are. I can answer that. We're working with Tracy Manick to investigate that exact association.
And I can supplement the answers by saying that what are we observing in terms of the type, we're looking at the type of preterm delivery and DNA methylation markers, and the markers we are identifying is different than the ones that are associated with positive health. Furthermore, in the positive health analysis, we do adjust for gestational age. And so it's very possible that they are all in the same pathway, yet they seem to be related to the outcome independent of gestational age and type of preterm delivery.
And one last question, Jackie, have you looked at folate levels in this cohort?
I'm not aware if we were able to assess folate levels. Rebecca, do you know if we were able to look at folate in the cohort?
We don't have folate levels, but Mike, I believe we would have information on whether or not a woman took supplements prenatally.
Yes, that's correct. Okay, well, we should move on. Thank you very much, Dr. Bangma. And
Thank
our
you.
next talk is by Heather Burris from the University of Pennsylvania. The title, The Promise and Pitfalls of Precision Medicine to Resolve Black-White Racial Disparities in Preterm Birth. So Heather, if you'll, there you go.
Yes, I'm getting there for some reason. I'm getting there. Let's see. I'm wondering, I had no problem. Oh,
There.
let's see.
Yeah, we did see it for a moment.
Yeah. Can you see this?
We do,
I'm gonna,
yes. Thank you.
there we go. Okay. And you can hear me just fine?
Yes.
Okay, great. So I'm going to talk a little bit about the promises and pitfalls of precision medicine, specifically when it comes to Black and white racial disparities in preterm birth. Because I often get the question, is it possible that, for example, DNA methylation or epigenomics could explain the differences we see between Black and white preterm birth rates? News outlets will ask me these questions, and to give a little bit of a spoiler, I think the answer is we don't know the answer, and I'm going to explain how and why we don't know this answer and what work might need to be done in order to answer this question. So I have no disclosures. So I'm going to talk a little bit about disparities overall. This group should know a fair amount about that already. And then I'm going to talk about some omic analyses that might be able to kind of answer these complex questions. Talk a little bit also about the problematic concept of biology of race, and then the implications of these types of analyses to answer this particular question. So we know that non-Hispanic Black infants die at twice the rate of Black or white infants in the first year of life. So overall infant mortality in the US, most recent data, it's about 6 per 1,000, and it's approximately 11 for Black infants and about a little under 5 for white infants. And what's striking is that among deaths, Black infants make up 29% of all deaths, whereas they make up only 15% of all births. So Black infants are overrepresented among infants that die in this country. And the relative risk of death by race varies by cause of death. Specifically, you can see that it is the preterm birth, low birth weight related deaths that are really disproportionately represented among Black infants, as well as pregnancy complications. These are the relative risks for each of them. So they're elevated for all, but most highly for the preterm related mortality. And then among infants that do die due to preterm birth or low birth weight, it makes up about 17% of all infant mortality, but 22% of Black infant mortality and just 13% of white infant mortality. And of all of the preterm related deaths in 2017, 37% were among Black infants. And again, this is interesting and striking because Black infants only make up 15% of all births. So there's overrepresentation of preterm related mortality among Black infants. And this is because Black infants are more likely to be born preterm. So whether we look at preterm birth at less than 37 weeks, whether we look at extremely preterm delivery, low birth weight if you don't believe the gestational age ascertainment, or very low birth weight, all of these measures are much more common among Black infants than they are among white infants. And I'll postulate to you today that genetics are not the driver of racial disparities in preterm birth, and I can talk for an hour about it, so I won't. But most of the studies of genetics of prematurity in general, separate from disparities, are done in homogeneous populations, like most GWAS or genetic studies. There are some small effects of single nucleotide polymorphisms with preterm birth within groups, but environmental factors play a much larger role when it comes to preterm birth, and specifically the difference between Black and white preterm birth rates. So I've started to think about health in general and racial disparities in health in general along a continuum, in part because I've kind of been on the side of this being environmental, and will sometimes get criticized for that, for not recognizing that there may be a small genetic contribution. And here's how I'd like to explain this. So along this continuum from genetic to environmental racial disparities in any health condition, we have our extreme versions of genetic racial disparities. These would be your sickle cell disease, your cystic fibrosis, your Gaucher's disease, where Ashkenazi Jewish populations have disproportionate risk. These are one gene mutation type diseases. Then we kind of move along the spectrum. We get to some of the cancers where there might be BRCA mutations or specific mutations involved in multiple myeloma that leave certain racial or ethnic groups at higher risk. But there's also a genetic or an environmental contribution. So perhaps a genetic predisposition on top of an environmental hit. And then we have our multifactorial heterogeneous disorders. Asthma, COPD, hypertension, and I would put preterm birth in this, where the disorder itself is heterogeneous. And this is what I was getting at a little bit with the question that I asked of the Elgan study, which is, is the phenotype, which even when we get to medically indicated or spontaneous preterm birth is quite heterogeneous. You're not going to have just one single nucleotide polymorphism that's going to be responsible for this heterogeneous set of phenotypes. And so the more heterogeneous the phenotype, the more genes that might be involved, the larger the environmental contribution often is. And so preterm birth is over here toward the right. And then we have our extreme only environmental exposures that are going to be responsible for racial disparities, gun violence, potentially car wrecks, depending on who owns cars, work-related injuries. If you're gonna die of a fishing accident and only white men are fishing, those are the men who are going to die. And so you're gonna have racial disparities or ethnic disparities based on just solely environmental. But I would put preterm birth way over here on the right-hand side. And a lot of our work has focused on that, but we have developed kind of an expanded framework for preterm birth risk overall, where we think about individual risk factors, maybe some of these genetic factors, and even the microenvironment. I mean, we know that smoking is a big risk factor for adverse birth outcomes, but a small number of women smoke. And really, when we think about population differences in outcome, we have to think about population differences in exposures. And so our group is really focused on some of these broader environmental factors that vary by population. So the air we breathe, the water we drink, the metals that are in our pipes, the social exposures to racism, discrimination that really vary by race and ethnic groups. So when we think about whether omics or precision medicine can kind of get at these factors, these are all these exposures that humans can have. And we then can, with our new fancy technologies, measure all of these multi-omics that Dr. Wang talked about. And you can see epigenome, transcriptome, metabolome, proteome, microbiome, there's now the immunome. I mean, there's omics everywhere. And the question is, can these exposures be kind of queried by using these omic biomarkers, and then maybe explain how these exposures lead to preterm birth as the outcome. And what I would say is that these fancy laboratory assays still are going to be sensitive to all the epidemiologic pitfalls that we have in any other type of observational study. So when we think about factors associated with self-identified race, they may be associated with these omic biomarkers, they may be associated with preterm birth, but these relationships may not be causal, and confounding will still plague our omic studies. So I'm going to give an example of this, walk through an example of how this may play out. So let's say our hypothesis is that DNA methylation differences by race explain a portion of the racial disparities in preterm birth. And say we hypothesize this because we observe that DNA methylation differs by race. And indeed, it often does. And one of the reasons it may is that you may have not even functional differences in SNPs, where there's a CPG site where you have an opportunity to have methylation that could vary by even ancestry, that can track incompletely with self-identified race ethnicity, which is a social construct, but nonetheless there can be a correlation with ancestry. And so we often see differences in DNA methylation by race that may be attributable to that, or may be attributable to other factors that are tracking but with race. And so the question then becomes, does race cause differences in DNA methylation, and maybe DNA methylation differences cause preterm birth? So is this kind of a causal relationship? But I would pause for a second and say, well, maybe, but there are many other possible explanations for this kind of observed mathematical relationship. So let's use air pollution as an example. This came up in one of the questions in the chat. So what if Black women have higher air pollution exposure than white women on average in this country? And the answer is they do. So this was a really elegant study. It's a modified figure from Tessem and colleagues, where on the x-axis we have year, and on the y-axis we have exposure to here PM 2.5, so small particles that are so small you can breathe them in and they'll cross right over the pulmonary epithelial barrier and get right into the bloodstream. And what we can see here is that the Clean Air Act has been associated with a decrease in overall exposure in the United States for everyone, but that we still see a pretty striking disparity where Black Americans have higher exposure than white Americans. And we know from even in controlled human experiments where Andrea Beccarelli's group has put healthy human volunteers into, I know this sounds scary, but into air chambers, basically transporting people to the levels of PM 2.5 you see in cities like Beijing. You're allowed to travel to Beijing, thus you're allowed to get into a chamber of PM 2.5. That would be the level of Beijing. And he and his team have shown in really elegant crossover studies that there are differences in DNA methylation after acute exposure to PM 2.5. So in this case, we've got these three pieces of evidence: DNA methylation differing by race, Black women having higher air pollution exposure, and air pollution causing lower DNA methylation. And so it's possible that instead of this direct association between Black race, lower methylation, and preterm birth, that perhaps we're seeing Black race causing, or being associated with where people live and being associated with higher air pollution exposure. There could be another unmeasured biologic process that has nothing to do with DNA methylation that is leading to preterm birth due to this causal relationship. Or perhaps there is some causal mechanistic relationship between DNA methylation and preterm birth, but it won't be the complete explanation. So there's a threat here that methylation is simply a biomarker or a passerby, but has no mechanistic role. There's another threat here, which is that air pollution itself has no impact on this outcome, but that Black race leads to exposure to racism in our country. This could change stress hormone responses. Maybe that changes methylation levels as well. Or the stress hormone itself is causing, or this pathway here is causing preterm birth, but that this arrow here with the question mark doesn't exist at all. And again, we're measuring methylation, but there is no actual cause of the methylation and preterm birth relationship. So in order to really get at this question, we need representation first, right? So we need to make sure that we have enough humans being studied in order to be able to ask and answer this question, even if we carefully control for all of the potential confounding variables and other pathways. And so our group last summer did a systematic review of all of the epigenomic studies of specifically DNA methylomic studies of preterm birth to look at whether there was adequate representation in order to even be able to answer the question that I get from the news outlets about whether epigenomics could explain racial disparities in preterm birth. And what we found was that because of folks like Dr. Wang, that there is in the US studies where race and ethnicity is specified, or actually global studies where race and ethnicity is specified, there is pretty good representation. So these were the number of deliveries that were studied with respect to epigenomics and preterm birth. There, however, were lots of studies where race and ethnicity is not defined, mainly because these studies are done in places like Norway and other places that are a little bit less diverse. And so there's kind of an assumption about what the race and ethnicity might be. Using population levels of race ethnicity, we were able to kind of extrapolate what the representation might be. And in this case, we have much lower representation kind of globally of Black women or African women or African women of African ancestry. And so while we had some representation when we talked about these original 16 studies, when we get to the entire set of 25 studies, there's less representation. But even if it were accurate, the primary problem is that almost all studies of preterm birth, epigenomic methylomic studies of preterm birth, are cross-sectional. Specifically, the samples are collected at the time of delivery. There were only 2 studies with 64 deliveries where maternal specimens were collected prior to delivery. The rest of the studies, therefore, suffer from potential reverse causation. So the cause of the preterm birth, so the example of preeclampsia or infection that could potentially lead to spontaneous preterm birth, might be causing the methylation changes that we observe in the placenta or in the cord blood or even in the maternal blood at the time of delivery. But that may not, those methylation changes might have nothing to do with the differences in preterm birth risk by race or ethnicity, or even in preterm birth overall. And also newborn samples may be simply documenting developmental stages. So some folks have done, Becky Simmons and others have done work where they collect samples of elective terminations in order to try to get at whether developmental stages are responsible for the differences we see in DNA methylation by gestational age. But it may be that as methylation changes over the course of development for tissue development, cellular differentiation, that's really what we're measuring when we see differences in a 23-week placenta compared to a 37-week placenta. So it may be normal and not pathologic. Okay, so how can omics help when representation is adequate and let's say we also have prospective studies? So I think they can still help us generate hypotheses regarding mechanisms. And this is specifically helpful when we have race stratified analysis. So when we're asking the question about disparities, if we see the same association, whether we are looking only among Black women, only among white women, or only among Asian women. And then we can look at different doses of exposure, and we can use causal mediation analyses to help us. And I'll give an example of that. And then lastly, we do need our mechanistic studies. We need friends in labs where we can perform experiments in order to really ask and answer the question as to whether DNA methylation or whatever omic signature you're looking at is causally related to the outcome of interest. So the other day I was running an analysis looking at neighborhood deprivation in Philadelphia. So I now live in Philadelphia, and for those of you who've been to Philadelphia, there's a fair amount of neighborhood deprivation across the city, but it's not uniform. So on the x-axis, we have neighborhood deprivation, which is an index that Cole Brokamp developed using American Community Survey data from 2015. So this is poverty, it's percent vacant houses, it's percent uninsurance, medical uninsurance, it's and median household income. There are 5 measures that Cole Brokamp uses. But what we can see here is that we have overlapping but distinct distributions, with non-Black women having much lower neighborhood deprivation, so less deprivation on the left side. And on the right side, we have Black women in Philadelphia with higher levels of neighborhood deprivation. And this is due to ongoing structural racism and ongoing residential segregation. And when we looked here with an omic signature, this here was actually the cervicovaginal microbiome, dichotomized into a high-risk microbiome, this CST4, which is dominated by anaerobes and has a paucity of lactobacillus. So that's our red dots here. We can see that a higher neighborhood deprivation index, we looked at everyone in this cohort, PI is Mikhail Elevitz. This is a prospective nested case control study. So there are 581 individuals in this analysis. And these samples were taken at 16 to 20 weeks of gestation. But here we can see that the cervicovaginal microbiome is more likely to be this high-risk microbiome if there's more neighborhood deprivation. And for a long time, people have noticed that African American women and non-Hispanic Black women have higher rates or higher risk of having this suboptimal neck microbiome, this anaerobic predominant microbiome. But we can see here that whether you're looking at all women, or you're looking just among Black women, or you're looking among non-Black women, that this high-risk cervicovaginal microbiome is more common with a higher neighborhood deprivation index. And when we looked at specifically here, this is the odds among Black women of having this high-risk cervicovaginal microbiome. You can see that indeed, consistent with the literature, Black women had about twice the odds of this high-risk cervicovaginal microbiome. But when we adjust for neighborhood deprivation, this association is diminished. And this is adjusted for multiple factors that I'm having trouble reading, but you might be able to. BMI, parity, insurance status, marital status, which is socioeconomic indicators. And so this would be kind of the older, more traditional way of doing a mediation analysis. Let's enter our mediator and see what happens. Here, my hypothesis was that it wasn't Black race per se, but environmental exposures associated with race that were really driving the differences in the cervicovaginal microbiome. But now there are newer ways to do causal mediation analysis. There are many people who work on this, but Tyler Vanderweel's group is really probably the leader of this. And in here, we can quantify the proportion of this total effect here of Black race on this high-risk cervicovaginal microbiome that might be mediated by our mediator here, neighborhood deprivation. And we can see this. This is the average causal mediation effect. This is the average direct effect of race alone. And then this is the total effect. And you can then get your 95% confidence interval. So there, here we can see that about almost 30% of the Black cervicovaginal microbiome relationship might be mediated by neighborhood deprivation. So these are the types of tools that we can use to start to disentangle whether an environmental exposure might be mediating the association we see between race and our omic signature. So if indeed our omic signature is not involved, however, at all in the causal relationship with, in this case, preterm birth. So let's say the cervicovaginal microbiome, even though it's high risk, is not really causally related at all to preterm birth, because we can't randomize women to a suboptimal microbiome at this point. There are some efforts to think about ways to improve the cervicovaginal microbiome and see if there are improvements in outcome. But at this point, we still only have observational studies. I still think that these omic signatures are not a total waste. There's still an opportunity for them to risk stratify individuals. So if we had a really good composite biomarker of all the potential risk factors that we already know, but we don't spend hours and hours interviewing women in prenatal care. So if we were able to get some sort of composite biomarker that would help us know which women are at highest risk of delivering preterm, perhaps this would facilitate enrollment into studies to prevent preterm birth. So this might be, we've done this before with only enrolling women with a prior spontaneous preterm birth. That's one of the highest, the strongest risk factor for recurrent preterm birth. And so in this case, we might be able to use some of these omic signatures to enroll women, targeted enrollment. It also might help us with regionalized perinatal practices. I mean, I came from Massachusetts, where there's really robust regionalization. Women are really kind of filtered into and funneled into hospitals that can best handle extremely preterm infants, and women are moved. And so you can imagine doing really great intrauterine guidance if we had a wonderful composite biomarker and could do better than just short cervix and fetal fibronectin, which both are suboptimal. The other thing I think is important is to get some scientific buy-in. So we know that exposures differ by race in this country due to longstanding racism and environmental injustice. But being able to document the biologic manifestations of this sometimes can be useful and powerful, but it does come with the risk of playing into kind of the biology of race, that there's something intrinsic or unmodifiable by race that has to do with something that humans are born with. And I think we really need to be careful and make sure that we understand it's not that the cervicovaginal, in that case, that the cervicovaginal microbiome is different because somebody's Black. No, it is because humans live in different environments and are exposed to really different stressors due to race in this country, due to ongoing injustice. So omics may be helpful to generate hypotheses, may be helpful for risk stratification. We have to be thoughtful about the consideration of these biomarkers when it comes to race. And instead consider the exposures that lead to the outcome. Ultimately, altering policy is going to be the main goal. But at the biomedical level, we still may be able to use these signatures to mitigate the biologic effects of exposures through interrupting these omic pathways, if indeed we can demonstrate that they are causally related to interrupt those pathologic processes, in this case, that lead to preterm birth. I'd like to thank you guys for listening to me, thank the organizers, our funders, and I'm happy to take any questions. And I couldn't see the chat, so maybe it's happening. I'll stop sharing.
Thank you so much, Heather. That was very provocative and interesting. And we have a lot of questions in the chat box, so we'll have to probably limit this a bit. Let's see, one causation study for the impact of air pollution on asthma was the Atlanta Olympics, where they were rerouting traffic for a period of time. Do you know if preterm births fell off in that interval?
Yeah, I'm aware of that study with asthma, which I think was a pretty acute response. I think with we've looked a lot at air pollution and preterm birth and others. I mean, we've looked at the literature and we've done some of our own smaller studies. A lot of these are latent effects, and they can take a long time. I don't think anyone published on the, I could be wrong, and maybe somebody on this call knows better than I do, but I don't think that the birth registry was queried in Atlanta with that same kind of natural experiment that happened. We're going to have another natural experiment right now with COVID because we know the air pollution levels have gone way down compared to what we have typically seen, just because people aren't commuting and flights aren't going and all of that. So I think there are going to be opportunities to look at that. I think it'll still be a challenge to get the right biomarkers if we care about how air pollution might lead to preterm birth, kind of going with the precision medicine.
Great, thank you. And then secondly, methylation in the scenario described is a response of the genome to stressors, social, environmental stressors. In this sense, how do we think of DNA methylation as causal, since it's not independent from the environment?
Yeah, so, I mean, maybe I'm not understanding totally. I guess when I think about it as causal, I mean in the causal pathway. So if you had your, let's use smoke. Smoking is a great example, right? So we know that smoking is associated with preterm birth. I think many of us think that smoking causes preterm birth in some women. We also know of all the DNA methylation studies, and I've done a bunch, and a lot of the people on this call have done a lot. I think the one kind of marker we can think about is that there's one CpG site that shows up in every single study of cord blood and smoking. And it's such a good marker that people have patented it for insurance companies to look for ex-smoking. But that doesn't necessarily mean that CpG site that is different in smokers or the offspring of smokers is responsible for the preterm birth. And I think people sometimes make that mistake. And I think I did when I was starting to learn about this. Instead, it may be that that's just off to the side. It's kind of a passive observer of what's going on. Yes, it caused that, but yes, smoking caused that change in methylation, but the change in methylation had nothing to do with the biologic process that led to the preterm birth. And maybe it is on the pathway. I just don't think we have enough evidence yet to say that those are causally linked.
Great, thank you. One last question. Apart from collecting placental samples prior to delivery, are there other methods you've considered to control for confounding by cause of preterm delivery?
Yeah, I mean, this is how so the placenta is going to be a problem when it comes to cross-sectional studies forever, right? Because we're not going to let people stick needles into women without a clinical reason in order to get a placental sample. So I've done some cervical studies. We've taken cervical swabs. These are still problematic because you can still, so prospectively, across pregnancy, Dr. Elevitz has also done that at Penn. But the trouble with those is that we still get this heterogeneous cell type. It still could be, there still could be other reasons that the cervix differs, recent intercourse, other types of co-infections, there are many other troubles. So I think if we're going, I guess I'm going through my, for any of the practicing physicians on the line, I feel like I'm, like a second year fellow where I feel like first year I learned so much and thought I knew so much. And then by second year, oh my goodness, there's so much I still don't know. So I feel like with methylation, I think I went through a phase where I thought this is going to explain a lot. And now I'm worried that there's so much more to learn and we need to, and I think with those types of omics, these omic signatures that rely on a particular tissue type at a particular time in development, we're going to have to be really, really careful about the conclusions we make regarding exposure and outcome relationships. I guess I'll leave it there.
Great. Thank you so much. Wonderful. So our last talk is Annie Behrens from the University of California, San Francisco, and her title is Cumulative Psychosocial Risk and Early Child Development, Validation and Use of the Childhood Psychosocial Adversity Scale in Global Health Research. And Dr. Behrens.
Hi, everyone. First of all, thanks so much to the moderators and also to the other presenters for some really interesting talks. So I'm going to be sharing some work. Hold on, let me get my screen shared. I'm going to be sharing some work that is from a study that I worked on several years ago at this point, but a study based in Bangladesh that based on a collaboration between researchers at Boston Children's Hospital, the International Center for Diarrheal Disease Research, Bangladesh, and the University of Virginia with support from the Bill and Melinda Gates Foundation. And our task was to kind of, in the context of a study that was looking at hoping to integrate a mix of kind of neuroimaging markers and some microbiome research and then behavioral and kind of test battery-based measures of child development, hoping to develop kind of markers of early profiles of risk of adverse developmental outcomes and to allow us to pick up children who are on a more adverse developmental path earlier. In doing so, we wanted to look at a range of risk factors that might be contributing to different outcomes that the study was measuring, including psychosocial risk factors. And my role was working on figuring out how to measure that in this study context. So I have no disclosures or conflicts of interest. I'll go through kind of a standard structure of background methods, findings, and discussion. So starting with the background, the study that this measure development was part of is called the Bangladesh Early Adversity Neuroimaging Study, led by Dr. Charles Nelson, Masood Alam at ICDRB, and then William Petrie at University of Virginia. We were working with initially kind of defined as a low SES sample in an urban, kind of a peri-urban slum area of Dhaka, about 240 participants, and then subsequently added some additional middle and high SES controls. And we saw children, we were kind of piggybacking after of some existing diarrheal disease research cohorts that were followed longitudinally. Those were birth cohorts from studies led by Bill Petrie at University of Virginia. So they had done kind of a population-based sample where they went door to door and tried to enroll all pregnant women in this neighborhood of Mirpur, and then followed women prenatally, and then followed their children from birth up through 60 months of age. So we were kind of piggybacking on those visits and adding these additional measures. So we were looking at, trying to look at sort of things that we might posit to be SES-related risk factors in our sample, and including non-psychosocial things. And I recognize that's a bit of a false dichotomy, but you know, they were already measuring things like microbiome differences, malnutrition, infection, which obviously are very influenced by psychosocial factors. But we're also interested in looking at directly at things like maternal experiences of stress, exposure to you know, maternal social isolation, child exposure to different forms of corporal punishment of more severe kinds, or measures of neglect. And then outcomes that were added in the BEAM study were mostly neuroimaging study measures, as well as some behavioral scales. I would say primarily the WIPSI has been the one we've most heavily used, the Wechsler Preschool and Primary Scale of Intelligence, mostly since it can be used more longitudinally across different ages, and because it had been, it had the most kind of, it had already been locally adapted by ICDRB researchers and other studies, and has some better validity data for Bangladesh. So my role was to work on the psychosocial measurement piece. Our participants, since it was a birth cohort, or we were piggybacking off 2 birth cohorts, all of our caregiver participants were biological mothers of the children. There was a mean of 4.3 years of formal education, and about 32% had no formal schooling. So we had to figure out how to measure exposures. You know, we were interested in a kind of sort of a questionnaire-based approach, but we had to administer all the questionnaires by interview. And about 84% of participants lived below the international poverty line. And there was a pretty even breakdown between boys and girls in the sample. We did our exposure, our CPAS work on children 18 to 60 months. The motivation for this, I think everyone's probably pretty keyed into this, but I think there's growing focus on child development globally. I think largely as you know, early efforts in global child health, I think rightfully focused on prevention of vaccine preventable diseases and child survival. And I think there's a growing emphasis and focus on the centrality of helping children thrive and preventing preventable forms of developmental compromise. And then also acknowledging this large body of work on the role of adverse childhood experiences and kind of cumulative stress and other psychosocial exposures and influencing child development. And then evidence that these sorts of exposures are quite common globally. There's a 2016 systematic review that estimated about 60% of children had either experienced personally or directly witnessed in the household physical, emotional, or sexual violence, not counting what we might consider corporal punishment in the preceding year. But I think in global health research, lack of sort of locally validated and appropriate measures to look at psychosocial stressors still poses challenges. So our task was to try to develop a measure like this. I'll kind of skip over this, but you know, I think the motivation for this is also embedded in this interest in the kind of biological processes that might link early stress to later outcomes, kind of linking environment to phenotype through biological processes. Thinking about what exposures have been studied previously in those working on things like adverse childhood experiences. You know, there was a very influential ACEs study, kind of, I think, early in the era of focus on this kind of cumulative childhood stress that looked at a subset of exposures, including physical, emotional, and sexual abuse, neglect, caregiver mental illness, domestic violence in the home, parental separation or divorce, and then incarceration and substance abuse among close contacts of the child. There's been a lot of work to kind of expand and adapt things that we consider when we're looking at ACEs, and I think a lot of critiques and fruitful work to improve these frameworks, and particularly looking at some exposures that are embedded in social inequities and structural inequalities in lower SES and racial minority communities. So we were looking for, you know, trying to measure something that had involved very sensitive topics. So trying to find an instrument that was acceptable to the participants, that didn't cause them distress, that was tailored to the context in terms of capturing the things that were salient in the study community, feasible, I think one challenge is there were existing measures that were being used in Bangladesh for many of the different subdomains we might be interested in looking at, like maternal depression or social support, but it was quite burdensome if you're interested in looking a little bit comprehensively at the social environment to administer separate questionnaires. So we're trying to see if we could create this kind of index or like consolidated scale, and then looking to generate kind of higher quality validation evidence than we had for a lot of the other tools available. As far as methods, we started with a kind of conceptual model generation, just looking at a literature review of the existing literature review of similar studies and similar measures. Then we did a kind of brief qualitative work. We did 10 in-depth interviews with caregivers, and then 8 2-hour focus groups, 4 with Bangladeshi field officers and 4 with caregivers, and then some content analysis and got some expert input, kind of identified these groups of exposures as well as specific examples that seemed salient in the study context of what to study, and sort of focused on things that were direct exposures to the child, and then sources of kind of psychosocial stress for the mother in this case or caregiver, and community factors like exposure to crime and violence, neighborhood deprivation, or experiences of discrimination. We then went through a process of generating items, doing some kind of cognitive interviewing, pre-testing with participants, and reducing or shortening the measure. So in generating items, we kind of looked across a large number of existing measures to see, to look at questions that have been used in other contexts. We didn't borrow any directly, but definitely adapted some from a range of other instruments, and also generated a number of new items based on the qualitative work or input from our collaborators. We pre-tested it and then piloted it with a relatively small number of participants initially, and used mostly classical test theory methods to kind of pare down the items, choose the ones that were performing well. And I can say a little bit about how we define that. So at the simplest level, we started by just looking at, this is actually an example from not items that ended up in the CPAS, but some other measures we were looking at, but it's the only graphic I had saved that captures the method. So starting with 61 items in a particular subscale, looking at Cronbach's alpha, and then just looking at simple item rest correlations, how each item correlates with the rest of the items in that subscale, with the thought that we should really be measuring a single cohesive construct within each subscale. And then we basically, in an iterative way, removed the items that didn't cluster with the others and came up with something that had higher internal consistency and was shorter. We did a little bit of work with item response theory, but at that phase, we had pretty small sample sizes. And then eventually we came up with the final set of items and then administered it in our larger sample. By the time of this paper, I believe we had 248 completed surveys. We looked at content validity with exploratory factor analysis, internal consistency, convergent validity in terms of correlation with similar or related measures, predictive validity in terms of the ability to predict or correlate with future child WIPSI scores. We would have loved to use more neuroimaging-based measures at this stage, but that data just hadn't been processed yet. And then retest, and inter-rater reliability, and then thinking about incremental validity, assessing what this measure has added relative to what we had before it. Sparse findings, so this is just an example of part of the exploratory factor analysis. We basically, at this point, ran an exploratory factor analysis on the items and looked to see that they clustered together well by subscale. They did. We did actually do some additional refining of the instrument at this scale and kind of drop some items that had very complex cross-loading and seemed to muddy the water in terms of measuring each subscale. So factor loadings less than 0.3 aren't shown here. And again, this is just one subset of the questionnaire, but it's a little overwhelming to look at all of it. Then looking at internal consistency, we had in the end, these subscales are seen on this side of the screen. There were 54 total items, and each of the subscales had relatively good internal consistency. Then looking at correlation, we had a number of other measures that we were administering in the questionnaire. We were using the home measure, which is the home organization of the environment, and that involves home visits, looking at different aspects of the child's care environment. We had the multidimensional scale of perceived social support, and the Edinburgh scale of postpartum depression that we were using, not just input, we were using in the entire sample. And there were reasonably good correlations, although not very strong ones, which was interesting. And that was kind of has led to a lot of contemplation for me. Then there are also pretty good correlations with raw IQ score at 60 months. So this would be comparing the 48-month CPAS score on each of the subscales with the 60-month WIPC score. Also, and this is sort of some very basic regression modeling that we really, I think at this stage we're sort of doing it as a form of predictive validity, saying does this measure correlate with the variables we would expect them to correlate with in the directions we would expect, and we found that it definitely did. This is just a multivariate regression model looking at adversity and then controlling for stunting status, which had been a big focus in the study as kind of a hypothesized predictor of poor development, and then controlling for an SES composite score that we developed that incorporated various aspects of parental income, parental education, different household assets and resources, and a number of other variables that I could share if anyone's interested. I think what we see is that the adversity score is quite strongly correlated, and that these models actually accounted for a relatively large share of variance in 60 months with the score. And interestingly, that the CPAS score accounted for, in doing kind of stepwise models that added variables one at a time, can estimate that the CPAS score accounted for about 20% of additional variance above the other predictors, compared to 9% for the SES measure and 8% for sensing. There could be many reasons for that. None of these the SES composite score could be an imperfect measure of SES, but it was at least kind of thought-provoking for us. So thinking about discussion, so the CPAS offers a single measure assessing multiple domains of psychosocial risk. It takes about 30 minutes to administer, and there were pretty strong psychometric properties in this initial study population in DACA. And it's available for adaptation and additional study context with some guidance on what kind of feasible and appropriate amount of local adaptation might look like. Currently working on this there's a global scale for early development, actually another effort to develop a global measure for child development outcomes that's being led by the WHO with Gates Foundation funding. We're looking to kind of piggyback the CPAS on this to validate it in some additional context to see if it can serve as a predictor of development outcomes. It's free and open source. So thinking about what adaptation might look like, I think, doing this project gave me a huge appreciation, I think, for both the challenge, but also the kind of central importance of measurement work. I think it's often, there's a risk of it being undervalued in research in general, but especially as we're taking these measures of very socially embedded experiences and constructs, and borrowing measures created in one context and applying them elsewhere. I think having some attention to this basic measurement validity is hugely important. So kind of what we've envisioned for a way to adapt it is, first do a basic review of the content validity and item validity with local collaborators, identify any items that they say, this definitely wouldn't work here, just take it off. If there's any content domains missing, add in a subset of items that you, working together with collaborators, you think would measure a needed construct. Pre-test it with parents. A number of about 25 in general, or even less, I think can be adequate, at least to identify any majorly problematic or confusing items. Any items that lead the participant to think of a totally different construct than you meant them to think about, and could be done relatively efficiently. And then piloting the item. One thing that we did in this study is that we used the data from the pilot sample in the final analysis, which I think is something that you'd have to justify in your analyses. But so I think to do classical test theory analysis and just look at item rest correlations and remove questions that did really poorly, you don't need a particularly large sample size. You can administer a slightly longer version of a questionnaire to the first 30 to 50 participants And then if you can justify it and be careful about it, you can still include their responses to the items that are ultimately included in the final measure. You can still include them in those analyses, so you don't kind of lose that pilot sample. You just have to account for maybe allowing a slightly longer amount of time for them to take a longer version of a measure. And then just reporting out some, do some basic validation analysis, looking at internal consistency and, depending on the resources and time and availability and feasibility, doing some basic test-retest reliability and correlation with any other similar measures that might be available. There are definitely limitations in this work, so there's still some gaps of validation steps that we would like to do. Looking at confirmatory factor analysis on the entire questionnaire. We've only done exploratory thus far, but we'd require a different sample to do that. Technically we shouldn't do a confirmatory factor analysis on a population that we already did an exploratory factor analysis on. Content gaps, we get a lot of questions about looking at protective factors in the environment, which I think is extremely important, and an interesting kind of ideological question of how we differentiate between protective factors and risk factors, but can only take on so much in one measure. But then, and also thinking about whether this measure would be appropriate for use with other caregivers. We were challenged by kind of a lack of gold standard comparators, so we found that correlations with other similar measures were only partial. And it was hard for us to say if that was because the other measure wasn't really measuring the construct that it was meant to measure in this setting, or ours wasn't. We have some reason to think that ours was probably capturing it based on comparative ability to predict WIPC scores. Then there are issues around the validity of the outcome measures. So the WIPC is a very culturally embedded construct. It was, I think it was a useful, at least to have something to validate the measure against, but I'll be interested to look at relationships between the CPAS with other outcomes. And then focusing on this one, I think there are generalizability concerns. So we did a lot of work to create this measure that works well in this specific population, but recognizing that it's going to take additional work to ensure that it's working well in new populations and that it has broader use. Thinking very quickly in the last 3 minutes about kind of whether all this extra work makes a difference. In general, when we looked at, so for instance, looking at the Edinburgh postpartum depression scale, which is a great measure for a lot of uses and is very useful clinically, and very widely used in research, has been previously used in Bangladesh with some kind of minimal validation evidence published. We found that while the CPAS depression subscale did predict scores on child development measures, the Edinburgh score generally did not. Which in my mind, if we're positing that those are both meant to measure similar constructs, it sort of suggests to me that there's a concern about whether the Edinburgh was really capturing the construct that it was intended to capture. Similarly, we did some early kind of like confirmatory factor analysis models on a small sample that we did have available to do that, and found that the Edinburgh items, the models initially fit the data pretty poorly. We removed one item that had low item rest correlations as if it was adequate. The depression subscale had good model fit for in the sample. And then in terms of relationship to WIPC scores in a measurement modeling context, again, the Edinburgh scale did not predict child development outcomes, whereas the depression subscale did. So yeah, just a plug for measurement work. I think that simple psychometric analyses of classical test theory statistics can help tweak an instrument. Even just, if you find one item that doesn't correlate with any of the others in a particular context, you can throw it out and make your measure maybe a little bit more valid. I think often there's not an expectation that we do that when we're using a measure in a new context, but I would argue that I would push for that becoming more and more the standard of practice. And then I think something we've seen in this context is that in initial analyses, I think there was an impression in our study that psychosocial variables just, quote, like weren't predicting anything. And I would argue that there's a risk that that's because of excess measurement error that we had introduced by using measures that hadn't been validated in the local context. And in turn, that kind of measurement error can bias what variables appear important and what we ultimately try to intervene on when we're trying to improve child development outcomes. In doing that, I think that can sometimes happen because of there's a risk of trading validity for comparability. This impression that if we use the Edinburgh, because lots of other studies have, that we're going to have results that are more comparable across settings. I think the concern there is when you're thinking about the fit of a measure to a particular context, you have to posit that it's basically a different measure as it operates in these different contexts. And that if you're giving up the kind of validity of the measure, it's hard to know what you're comparing. And then, yeah, that's all. So many thanks to many collaborators. There's much work still to do on this. Right after I finished this paper, I started residency at UCSF and have 2 little kids, and my work on this project has completely stopped. But in about 3 weeks, I'm starting fellowship in developmental pediatrics at Stanford, and we'll have a lot more research time. So I hope to pick this back up again and look at relationships with some of the neuroimaging outcomes with some, more well-developed statistical techniques. Thank you very much.
Thank you so much. Beautiful work. And we have just a few minutes for a couple of questions, and then Dr. Bear wants to moderate the final discussion. So let's see, Dr. Behrens. One question is, how does the CPAS overlap with the positive child health index? I guess asking whether this might be a tool that would be predictive of a broader range of outcomes beyond cognitive outcomes. Any speculation as to whether it might predict behavioral outcomes or asthma, obesity, that sort of thing?
Yeah, I think it's something that I would love to look at in the future. And my hypothesis would be that it would. It's been very predictive of most things that I've plugged into kind of just basic exploring correlations. We had some maternal inflammatory marker data and plugged the CPAS in to see if there was a correlation, and there was. And that so I anticipate that it probably would correlate with a range of later outcomes. Again, that by no means proves that that's a causal relationship. Both the child health outcomes and the exposures in the CPAS are influenced by potentially similar underlying structural or environmental variables. But I think that's where having some of the things like neuroimaging, maternal inflammatory markers, microbiome data to try to start to at least posit and model out some proposed causal pathways is exciting.
Yes, thank you. And then one other question is, can you do something like a 10-fold cross-validation if you don't have a validation cohort?
Not sure I understand the question.
Maybe Dr. Burris, do you want to unmute yourself? And
Sure. Yeah, I was just asking. I mean, it seems like you've done all this work, and then you said that you would need to look at it in a separate cohort. But I wondered if there's a way, and maybe that's what you did here. But it looks like these were all kind of individual correlations. I wondered if you kind of used your new measure and kind of kept in a bootstrapping way, or in a way to be able to kind of resample your cohort. Would you get the same answer is I guess the question.
Yeah, it's an interesting question. Not something that I've done, but I think it would be worth doing. We also have a lot more data at this point. The study's been ongoing in my absence, so we may have the additional sample at this point to use.
Okay, well, again, the presenters, beautiful work, and it's great to see a lot of young people presenting. And Cynthia, let me turn it over to you.
Thank you, Mike. This has been a really very interesting plenary session, and I want to thank all our speakers. I think we have maybe like 5 minutes for general discussion, and I kind of want to kick it off by saying that I see some common themes between all 4 of the talks, and maybe in some ways that they can inform each other, such as the use of folate or choline in the studies looking at DNA methylation, and if that's a factor in the last 2 studies and an indicator that should be looked for. as well as the idea that maybe some of the outcome measures like the CPAS and the predictive child health outcomes indicators, if I have that right, how they could actually, is kind of a theme that runs through all 4 talks. And I wondered if one of the speakers could comment on maybe either the folate or the additional use of other outcome measures that might be available from the research being presented in this group.
I don't know if Rebecca is still on, if she might want to comment on folate as an intervention.
So at least in Project Viva, a lot of the maternal intake of methyl donors didn't quite pan out the way that we were hoping they would, or the way that they do in the agouti mouse model. I think in the early days of this, we were all hoping that, you know, we could solve these large heterogeneous problems with a dietary supplement. And I've fallen into that path. I mean, I've fallen into that. I've stumbled upon vitamin D and stumbled upon folate, and I think that it's not as simple as that, and I don't mean that in a pedantic way, but I think we're dealing with so many co-exposures all at once, and there's probably, like anything else, an adequate amount of folate that's needed for optimal health, and maybe a little bit too much is maybe not great, but that I don't think that we have the tools yet to say this exact amount of folate is going to affect the DNA methylation of this particular gene, this amount every single time. I mean, I feel like I worry about our micronutrient. I worry about our micronutrient pathways as the answer to our overall environmental exposure problems.
As a former enzyme kineticist, I agree. I don't think that just filling up your tank with folate changes the regulation of those pathways. And probably it's the regulation of those pathways that needs to be affected. Maybe
I also think about, it also makes me think about common dietary sources of folate and how that's related to access to leafy greens and other social environmental variables. And the fact that folate is a recommended prenatal vitamin are people who are able to regularly access prenatal care and have good health insurance and less chaotic life situations, able to follow that recommendation more.
So I'd like to add to what has been said. And so I agree, folate, on the one hand, is extremely important. So actually, folate is only one micronutrient currently as a part of the mandatory fortification. And this, aside from vitamin D in the milk. And also, but we know a lot about folate, but still a lot of things remains unknown. So I wonder, perhaps you have seen the recent NIH release the precision nutrition roadmaps for the 2020 to 2030 is a 10-year roadmap. I wonder anyone have seen this. So I think if you haven't seen it, I feel it's worth to reading. I think it's outlined very exciting vision and to trying to have a better understanding individual variations, particularly when, where, and why a nutrients are affecting health. And it's underscore the vision, underscore individual variations, and also the timing. It's at the different developmental stages, different health status, and also different genetic susceptibility. And also, I think over the year we have learned for the folate, even folate is a beneficial micronutrient, essential nutrient. It could, at the two end, could also has a problem. Deficiency absolutely is a problem. If it's excess folate, could also, I think people also raise that concern. So how to optimize nutrition, that means not too much, not too low. So that is the part of the precision nutrition. It's at the individual level, at the different health condition, at different developmental stages, and the different people may have different requirement. And relate to that, I think my other colleague also emphasized the co-exposure, the mixture, nutrition, toxin. I think we have done several studies demonstrated the interplays of the environmental toxin and the folate. For example, we demonstrated folate potential here's counteracting smoking. We also published on counteracting lead exposure, counteracting with mercury exposure. So indeed, I feel this area has a tremendous potential, but they need a lot of work to have a better understanding before it become a prime time intervention. But I think a nutrition compared to a lot of other pharmaceutical intervention, compared to other more invasive information, potentially if we can get it right, it's relatively simple, scalable, and a safe intervention.
And I think that remark should conclude our session. Beautifully put. It's 1 o'clock now. And I think if we have further discussions, they should probably happen offline. The speakers have all given their emails on the chat box if anybody wants to approach them directly. And I'd really like to thank everybody for participating in this talk. I guess it's afternoon some places and morning other places. So have a good rest of your day, everybody. Bye-bye.
Thank you.
Thanks.
Thanks to all.
Thank you all.
Bye.
Thank you.
Bye.
Hi, thank you.